The Enterobacteriaceae family shares several important general features. Most members are gram-negative rods. Many are motile with Paratrichous flagella. They all ferment glucose and typically reduce nitrate to nitrite. Most are oxidase-negative. Antigenic structures are used for typing. The H antigen represents flagella. The K antigen represents the capsule or envelope. And the O antigen comes from the cell wall lipopolysaccharide. All Enterobacteriaceae possess endotoxin in their LPS, while some also produce exotoxins. Now let's highlight specific members. Escherichia coli is indole positive and lactose positive, with an IMV-IC pattern of ++-. It is the most common cause of urinary tract infections in females. The enterotoxigenic strain, or ETEC, produces both heat-labile and heat-stable toxins. The entero-invasive strain, or EIEC, penetrates epithelial cells of the large intestine and may be lactose-negative. The enterohemorrhagic strain, EHEC, especially serotype O157:H7, produces a Shigella-like toxin. It is associated with undercooked hamburger meat, food poisoning, and can lead to hemolytic uremic syndrome, or HUS. Additionally, certain K1 strains of E. coli can cause neonatal meningitis. Shigella species are lactose-negative, non-motile, and anaerogenic. They cause bacillary dysentery by penetrating epithelial cells of the small intestine. Among them, Shigella dysenteria group A causes the most severe disease. Shigella flexneri, group B. Shigella boydii, group C. Shigella sonnei, group D, which is the most common. Notably, fewer than 200 organisms are needed to cause disease. Klebsiella are opportunistic pathogens that can cause urinary tract infections and pneumonia. They are often resistant to ampicillin. Klebsiella pneumoniae is non-motile, has a capsule, and may cause lobar pneumonia. It is variable for urease, negative for ornithine, and positive for Vogus proskauer. A newer species, Klebsiella granulomatis, was formerly called Acinetobacterium and is known as a sexually transmitted infection. Enterobacter species are motile and ornithine positive. Serratia species may produce a characteristic red pigment. They are D-nase positive, gelatinase positive, and Vogus proskauer positive. Klebsiella pneumonia is a major opportunistic pathogen. Its most recognized role is in causing pneumonia, often lobar pneumonia, but it can also cause infections in wounds, the bladder, and the kidneys. A key feature is that it is non-motile. It lacks flagella and cannot swim. Biochemically, it shows a distinct profile. It is citrate positive, turning blue, lysine positive, urea variable, sometimes turning pink, and ornithine negative. Together, these traits help distinguish Klebsiella pneumonia from other Enterobacteriaceae in the laboratory. Several types of selective and differential media are used for isolating gram-negative organisms, particularly Enterobacteriaceae. McConkey agar uses lactose as the differentiating agent and crystal violet with bile salts as selective agents. There is no hydrogen sulfide indicator. Colonies that ferment lactose appear dark pink, while lactose-negative colonies are transparent. Eosin methylene blue agar, or EMB, differentiates based on sucrose and lactose. Selective agents are eosin Y and methylene blue. Again, no hydrogen sulfide indicator is included. Lactose fermenters may show a green sheen, purplish or brownish amber colonies, while lactose-negative colonies remain transparent. Hectoenteric agar, or HE, uses lactose, sucrose, and salicin as differentiating agents. Bile salts are selective, and sodium thiosulfate serves as the hydrogen sulfide indicator. Lactose fermenters form salmon-colored colonies, while non-fermenters appear green to blue. If hydrogen sulfide is produced, colonies turn black. Salmonella Shigella agar, or SS, uses lactose as the differentiating agent. It contains brilliant green and bile salts as selective agents. Sodium thiosulfate is the hydrogen sulfide indicator. Lactose-positive colonies are red, while non-fermenters are transparent, sometimes with a black center if hydrogen sulfide is produced. Xylose lysine deoxycholate agar, or XLD, differentiates using lactose, sucrose, and xylose. It contains bile salts as the selective agent, with sodium thiosulfate as the hydrogen sulfide indicator. Lactose-positive colonies are yellow, while lactose-negative colonies are transparent on red medium. Hydrogen sulfide-positive colonies appear black. Finally, enrichment broths such as selenite broth and GN broth are used to enhance recovery of gram-negative organisms. Salmonella requires a large inoculum to cause infection, usually more than 100,000 organisms. Biochemically, it is hydrogen sulfide positive, lysine positive, indole negative, and urea negative. Within Salmonella enterica, there are more than 2,000 serotypes. Salmonella cholerae causes septicemia. Salmonella typhi causes typhoid fever. Blood cultures are usually positive in the first week, while stool cultures become positive in the second to third week. Salmonella arizonae is unique for being ONPG positive, unlike other Salmonella. Citrobacter is an opportunist that is biochemically similar to Salmonella, but it is lysine negative, which helps differentiate it. Proteus species are notable for being urea positive and deaminase positive. Proteus mirabilis is indole negative and the most sensitive to penicillins. Proteus vulgaris is indole positive and hydrogen sulfide positive. Yersinia species include Yersinia enterocolitica, which grows best at room temperature and can undergo cold enrichment. It is invasive and toxigenic. Yersinia pseudotuberculosis, which causes acute mesenteric lymphadenitis and produces pseudotubercles. Yersinia pestis, the agent of bubonic plague. Finally, Pseudomonas is oxidase positive, now reclassified into the Enterobacteriaceae. It has Lophotrechus flagella and is associated with diarrheal disease. The oxidase test uses tetramethyl p-phenylenediamine dihydrochloride as a reagent. A positive result appears purple. The nitrate test uses alpha-naphthylamine and sulfonilic acid as reagents. A positive result is pink, but zinc powder is used to confirm true negatives. The ONPG test detects the enzyme beta-D galactosidase. The reagent is O-nitrophenyl beta-D galactopyranoside, and a positive result is yellow. The triple sugar iron agar test, or TSI, contains a small amount of glucose and larger amounts of sucrose and lactose. A yellow butt means glucose fermentation. A yellow slant means lactose or sucrose fermentation. A red slant means neither lactose nor sucrose fermentation, and a black butt indicates hydrogen sulfide production. The Kleigler iron agar, or KIA, is similar but contains only glucose and lactose with no sucrose. The citrate test starts with a green medium. A positive reaction turns the medium blue. Decarboxylase tests measure the ability to decarboxylate amino acids. Lysine produces cadaverine, ornithine produces putrescine, and arginine produces putrescine. The indicator is bromcresol purple. In lysine iron agar, LIA, a purple butt indicates positive lysine decarboxylation. A red slant shows lysine deaminase activity. In motility indole ornithine medium, MIO, a purple butt means ornithine decarboxylation. This test also checks for motility and indole using Kovacs reagent. The indole test detects indole split from tryptophan using Kovacs reagent. A positive result is pink. The urease test detects hydrolysis of urea to ammonia and carbon dioxide. A positive result turns the phenol red indicator pink. The Voges-Proskauer test detects acetoin, using potassium hydroxide and alpha-naphthol as reagents. A positive result is pink. The methyl red test checks whether glucose metabolism produces stable acids such as lactic, acetic, or formic acid. The oxidation fermentation test, or OF test, distinguishes whether carbohydrates are oxidized or fermented. The media is semi-solid with bronthomol blue. If both tubes turn yellow, the organism is a fermenter. If the open tube turns yellow but the closed tube stays green, it is an oxidizer. If both tubes remain green, the organism is a saccharolytic. Finally, the phenylalanine deaminase test uses ferric chloride as the reagent. A positive result produces a green color. Let's review the important gram-negative non-fermenters. The most common is Pseudomonas aeruginosa. This organism is an obligate aerobe and is oxidase positive. It grows on MacConkey agar but is lactose negative. It is an oxidizer, meaning it is positive in the oxidative fermentative glucose test. It is susceptible to polymyxin B. Pseudomonas is an opportunistic pathogen and is often mucoid in cystic fibrosis patients. It produces distinctive pigments: pyocyanin, which is blue-green, pyorubin, which is rust-red, and pyoverdin, which fluoresces yellow. It is frequently drug-resistant and causes infections in burn wounds, pneumonia, swimmer's ear, and the eye. The second most common non-fermenter is Acinetobacter baumannii. It is also an obligate aerobe. The bacteria may appear as bacilli or coccobacilli. It is oxidase negative, grows on MacConkey agar as lactose negative, and is an oxidizer in the OF glucose test. Another species, Acinetobacter lwaffi, is non-saccharolytic, meaning it cannot utilize sugars. All Acinetobacter are non-motile. The third most common is Stenotrophomonas maltophilia. This organism is oxidase negative and an obligate aerobe. It grows on MacConkey agar and is an oxidizer. It is of glucose positive and a strong oxidizer of maltose. The OF maltose test is positive. Finally, Burkholderia cepacia is another important non-fermenter. It is weakly oxidase positive, an obligate aerobe, and grows on MacConkey agar. It is an oxidizer in the OF glucose test but is resistant to polymyxin B. Burkholderia is an opportunistic pathogen, particularly dangerous in cystic fibrosis patients, and produces a yellow-brown insoluble pigment.